Hi, I'm Dr. Beckham and I'm the research educator for Virtual Cures, also known as Virtual Drug Screening. We want to take you through a walkthrough of the lab and show you some of the equipment and the resources that we have in lab and introduce you to a few of our researchers in there. First thing though is to get a little hand sanitizer. Now I've got masked up, I'm ready to go into lab. So this is our lab space that we share with the Aptamer stream. We've got lots of different pieces of equipment here, lots of different fridges. We have these bench tops that are low down that we can work on and we can see each other and communicate pretty well. Our goal in virtual drug screening is to discover new inhibitors against infectious diseases. So we use both wet lab and computational techniques to do that. One of our goals is to make the genes and DNA that we're going to use for cloning our proteins, and then expressing those proteins in bacteria and testing them out against our drugs. Let's do a walkthrough of lab now. So Mengzi is one of our researchers in the FRI stream, and she's working on some computer work. Mengzi, do you want to share what you're doing?
I'm currently working on some molecular docking stuff, and I'm just about to submit my job to the survey.
Awesome. So we use a multiprocessor computer cluster called the BRCF in order to run our virtual screening jobs on the computer. Thanks, Mengzi.
Thank you.
Going back here further, we've got lots of different machines. A lot of these are centrifuge machines for spinning our samples down when doing purification. We've got PCR machines for synthesizing DNA when we're doing our PCR work. Come back here. Kinsey, would you like to tell or guess what you're working on?
I'm making my agarose gel to run my DNA in for later on.
Okay, great. Thanks, Anne. So let's keep on going back. Some more PCR machines. We have our standing benches where we do most of our sterile culture work. And Jordan is one of our ARI researchers. And Jordan, would you like to share what you're working on?
Yeah, I just expressed my protein and I'm moving towards purification.
Awesome. So growing up big batches of protein, which eventually will get stored in our freezers here. We have a -20 freezer, and then also our deep, kind of what we call the deep freeze. This is a -80 degree freezer, and very cold for long-term storage. So let's keep on going back. This is a shaking incubator where we'll grow up our bacterial cultures. Right now there's no cultures growing in here, but we grow these up overnight. And then, John, you want to come over here?
Yeah.
Introduce yourself. John is one of our mentors. And John, what have you been showing the students?
Today I've been helping them make agarose gels for their DNA samples and doing some stock dilutions.
Okay. Sounds like fun. Thanks, John.
As always.
So let's keep on going back here. Back here, Marie is one of our ARI researchers.
Hi.
And Marie, do you want to share what you're working on?
Yeah, I'm just working on sonic-gating cells. We can break them open and get to the protein that we expressed.
Awesome. Thanks, Marie. So let's come on back here. This is our agarose gel area where we run our DNA gels in order to see if our cloning or PCR procedures have worked. We'll make an agarose gel here. We'll add some ultraviolet dye, and we'll throw them on these ultraviolet imaging stations, and we'll be able to see whether or not the banding patterns are indicative of whether we've gotten our cloning procedure to work. So this is the beginning of a long process that students will work on for many weeks until they get to the protein expression steps, growing up protein and spinning down and using purification strategies like these large centrifuges. But in the end, we hope to clone to plasmid DNA and then made protein and tested that protein against the drugs that we find in virtual screening. We hope you've enjoyed the lab tour, and good luck in your choices in the streams. Thanks.
Bye, Dr. V.